Hello, everyone. Thank you for coming to the PIBBSS Symposium 24. The PIBBSS Symposium is an event that finalizes the PIBBSS Fellowship and where the PIBBSS Fellows who have been working on the project for the last 3 months can have an opportunity to present their work. So our first speaker today is Clarke, who has been studying the patterns of co-occurrence in features in neural networks that are found by sparse autoencoders. And he will tell us about his work. If you have any questions, please use the Q&A function. The talk will probably last about 45 minutes, and there will be about 15-minute Q&A at the end. And if you'd like to chat with the panelists, you can do so at the end of the 3 talks. We'll share some meeting rooms for you to use. And Matthew, you can begin.
Right. Thank you very much, Mateusz, for that introduction. And let me just get a laser pointer up. Yes, so I'm Matthew. My mentor here at PIBBSS is Joseph Bloom, and today I'm going to talk about our work together on looking at co-occurrence in sparse autoencoded features. So I'm going to start with some background and motivation, and I think in the spirit of PIBBSS, I'm going to start with a really wide view. So PIBBSS stands for the Principles of Intelligent Behavior in Biological and Social Systems. And I think that the work I'm going to talk about today actually can kind of span all of those domains a little bit. So first of all, I'm going to talk about kind of the problem. And I think the problem is that human beings are threatened to some degree by a lot of different kinds of autonomous systems. Some of these are very basic. A tiger is quite scary when you don't control it. But also things like, say, pathogens. So, for example, at the moment in the United States, there's an outbreak of H5N1, currently only in dairy cows, but we can't control whether or not it will remain in dairy cows as much as we would like. But there are other kinds of autonomous systems we have to worry about as well. I'm going to make the very broad argument that society counts as one of these. An example of this might be nuclear early warning systems. These are systems that are created by human beings, but not wholly under the control of human beings. And they don't work as well as we would like. A classic example of this is the case where there was a incorrect early warning of a US attack on Russia. This was prevented by Stanislav Petrov. But before everybody gets too reassured by that, we must remember that Stanislav Petrov was demoted because he didn't pass on the information correctly. And then, most relevantly today, we are entering an era where there's lots of computational systems that are not 100% in our control and that are quite dangerous sometimes. Early hints of this are things like Tesla Autopilot, which doesn't work as well as we would like and causes crashes. Also, another kind of crash are stock market crashes caused by stock market algorithms. So there are all these different kinds of problems that we have to worry about. And I would argue that these kinds of autonomous systems actually can be lumped into a slightly smaller category, which are network-based systems, be these networks of neurons in a brain, or genes in a cell, or artificial neurons in a large language model. And within that very broad area, today I'm going to talk about AI safety and very slightly about biological safety. Both of these are concerned with kind of understanding and dealing with these network-based systems and the emergent properties of them. And if you want to be able to guarantee or at least have a good guess at whether or not these systems are safe, you need to have, in my opinion, some kind of mechanistic interpretability of what exactly these systems are doing. I'm going to define that slightly more specifically in a moment, but that's important both in AI and in biology. Within that problem of mechanistic interpretability, we have the problem of feature extraction. In theory, if you have a really big matrix of lots and lots of, say, gene expression, you might be able to train an algorithm to predict what's going to happen next, but I wouldn't say that you have an understanding of why that thing is going to happen next. To do that, you need to find some smaller set of features that you can extract from all of those data that are something that a human being can get their head a little bit more around. But that problem is quite hard. What sort of features do we want to extract? What are going to be best? A recent development in machine learning that is quite promising for the case of large language models is that of sparse autoencoders, which I'll explain even more in a moment. And in particular today, I'm going to be talking about a particular phenomenon of the kinds of features that you get with these sparse autoencoders, which is their co-occurrence on tokens within a model, and what we think that means. So to start out with mechanistic interpretability, I'm going to defer to a definition proposed by Lee Sharkey, which is that what you really want in terms of mechanistic interpretability is to have some kind of a mathematical description of the system that you're modeling that you can then use to, in a nice defined way, turn into features with semantic labels that you can use to understand what these functional parts do and how they interact, and that those features and their semantic labels must be good enough that you can make predictions of what will happen to this system if you observe a particular feature or if you activate a particular feature that you can then validate in the real world. And of course, these validations are not always going to be perfect, but you can hopefully use them to improve your mathematical description and do better. And this kind of mechanistic interpretability is going to be important in the kinds of safety problems that we're interested in, because it might allow you to understand why the model is behaving a particular way from its internal components. And then in turn, that might allow you to improve its capabilities, which of course some people would be against, but also fix bad behavior and maybe even predict bad behavior from the general setup of the model without necessarily running it, rather than having to let it go through some big gauntlet of specific cases and then worry that it might be able to slip through the cracks of those. So if we want to do this kind of mechanistic interpretability, we need to work out what a good functional unit is. And it would be lovely if the neurons in a machine learning model, in a large language model, were monosemantic and easily interpretable, if we could just find the dog neuron. And indeed, they're not imperfect in that we can decompose them into useful circuits, we can label them a little bit, but the vast majority of these neurons don't seem to relate to a very human concept. In fact, they seem to turn on for all kinds of very strangely unrelated things. So we need to find a way to convert these neurons into some other representation that is maybe easier to understand. And a hint as to how we might do this can come from why we think it doesn't work just at the neuron level, which is that neural networks are able to process a huge range of different inputs and produce quite subtly different outputs. And they're able to do this even though they don't necessarily have all that many neurons. So English has 10 to the 6 words, GPT-2 XL only has 307,000 neurons, but you can still get some pretty good sentences out of GPT-2 XL. And large models are very, very capable of very subtle distinctions. So, for example, they can tell the difference between visiting the tiger woods, which is quite dangerous, and visiting the tiger woods, which would be quite exciting, and you might get an autograph. And so, because of all of these features, all of these concepts having to be compressed onto this small set of neurons, we expect these neurons to have a property called superposition, where basically they are creating a vector space with more dimensions than you have neurons. And so each neuron is representing multiple features within that space. And so basically this means that your neurons, you would expect to get this property of polysemanticity from this superposition. And so our problem becomes all of these neurons are going to be representing things in combinations rather than as single neurons. And how do you turn those combinations back into a single property? And to make a biological analogy, when visual neural networks in real brains were first being studied, and these actually, of course, inspired a lot of the work on machine learning neural networks, we found that the first layer does very basic decomposition of your image into maybe dark patches and light patches. The second layer then does something a little bit more specific. It maybe finds where the edges are, and the next layer finds where the corners are. And people joked at the time that maybe if you just find enough layers, you'll find at the top something so hyper-specific that you could find a single neuron that encodes your grandmother. And this is my own grandmother. But of course, that isn't how these systems work, for the same reason as we don't get that in machine learning neural networks, because we can recognize a lot of people and a lot of concepts. But really the problem is, how can we artificially make a grandmother cell? So one potential answer to this is sparse autoencoders. So sparse autoencoders consist of taking your machine learning model, which has lots and lots of layers of different sets of neurons, and replacing one of those layers with a sparse autoencoder, taking out the activations that lead into it, splicing them into a single layer of neurons, ideally one that has more neurons than would be in the layer that you're studying, and then training this whole set such that you can take the signals from this large layer and squeeze them back into your machine learning model in such a way that it doesn't tend to change its behavior too much, that there isn't too much of an error from having this sparse autoencoder. But the advantage of having this very big layer in the middle rather than your normal neural network layer is that we can train this to be, well, sparse. That is to say, we can set it up so that hopefully only one neuron fires in this big layer at a time. And then these we expect to associate with nice, discrete, independent, hopefully, features. Now, we can't just replace all of our machine learning models with this because these things are quite expensive to train, but they do seem to work very well, which is very exciting. Now, something that I'm going to show a lot of diagrams from today is this website called Neuronpedia. Basically, this has a big set of sparse autoencoders for lots and lots of different models. And it allows you to look at a particular feature and try to understand what exactly is happening. Where by a feature, I mean a single neuron activation within this sparse autoencoder that corresponds to some pattern of neurons within the real model. And so for a particular feature, you might see what kind of tokens it promotes within a string of text, what kinds of tokens it prevents from firing in a string of text, and examples of strings of text where this feature has turned on and where it is most active. And so in this particular case, I've looked at a very small sparse autoencoder, so it doesn't have very many neurons in this big layer that it's training. And here I can look for things that activate when I ask it about things to do with days of the week. And I found a particular feature that turns on a Wednesday, but also on a Friday and on a Monday. So it's detecting weekday-ness in some degree. But with a small SAE, you can't find distinct individual features. And this kind of makes sense because the more neurons you have to train, the more fine-grained you can get about picking out these features. So in a larger SAE, now we can find, for example, a Saturday feature and a Wednesday feature separately. And how much you need to make your SAE large, what kind of things change as you go between these sizes is quite an important thing to understand because, as I said, training these is very expensive. And if we could get away with smaller SAEs, that would be really nice. So SAE features are a good step towards mechanistic interpretability because they're very clearly tied to their original model and quite simple in a way to understand how they come out of this original model. We find when we look at these, and there's been a lot of work from Anthropic, for example, on this, that they have quite often nice, clear semantic descriptions. So, for example, the Golden Gate Claude model so for example, and also they can be steered on. So if you activate a particular feature like the Golden Gate feature, then you get a model that talks just about the Golden Gate Bridge, and this is the Golden Gate Claude model. So they kind of fit into our 3 steps here. However, there are still problems with them. So SAE features can still be quite hard to interpret. And one of the ways that they become hard to interpret is they can often be quite similar and seem in a way to be redundant. And possibly this is just an artifact of the fact that we're training very large SAEs. So for example, here we have 2 features that both fire on the token how within the text. And they both promote relatively similar things. So they both promote how be it. This one promotes how much and this one promotes howsoever. It's not easy to see from this what the difference between these are, what kind of function they might be having within the model. And of course, we could steer the model on them and see what happens, or feed in lots of new prompts to try and work out what's going on. But of course, that involves a lot of running of models, and running models is expensive. And doing that for lots and lots of features would be expensive as well. So we would rather have some way of extracting some more information about the difference between these 2 features from kind of static properties that we can generate at the training stage of the SAEs. Another problem, or perhaps an opportunity, but certainly something that is very interesting, is that SAE features don't seem to be as linear as you might expect. So it would be very nice, again, if different SAE features could just be added up together, and the only information that they encode is how much this feature is present, so that you can think of them as scalars. But some work from Engels et al. And the Max Tegmark lab shows this isn't the case. They pulled out features corresponding to days of the week, and they performed a principal component analysis on cases where those features were active. And what they found was that the activation of the neurons, if you look at that in a 2D slice, seems to form this ring that correspond to the days of the week. And these days of the week form this ring in the order that you would expect. So it goes Monday, Tuesday, Wednesday, Thursday. Actually, it goes the other way around, but you see what I mean. And the hypothesis as to why this is happening is basically that the model needs to be able to do kind of a mathematical operation on these features. It needs to be able to say, well, what's the 32nd Tuesday after Sunday, or the 32nd day after Sunday? And to do that, it finds Sunday and it just goes round 32 times and then goes, oh, it's over here. It's a Friday. And so you have this information that's encoded in the direction of these features as well as just their magnitude. And you see this as well with months of the year and maybe even years in the 20th century. Another thing that we need to be aware of is that the sparse autoencoder is kind of imposing its own view in a way on how the model is working. So a sparse autoencoder is not going to find a compositional code. That is to say, if it wants to find things in this ring of weekdays, it's just going to find individual days of the week because our sparse autoencoder is trained to find single features at a time. So it's not going to provide a compositional encoding. But that doesn't mean that's how the actual model is doing this. In fact, there are lots of good reasons to think that the way that a model might be eventually learning to represent days of the week might be something more like this, where it goes to a sort of weekday subspace and then just uses numbers to find which day of the week it is. And a reason that you might expect this is that models are quite good at speaking lots of different languages. And so you might have a different weekday vector for different languages, but then you can reuse your number vectors to find kind of the lookup of, well, what is a Monday in German, for example? But there might still be cases where this compositionality is so strong that a sparse autoencoder can't decompose them fully and still gives us a little bit of a hint of this underlying compositional code. And the way you would expect to see this is that you would find features that aren't acting independently and can only be interpreted as a unit. And again, a reason that we might expect this to be possible is that sparse autoencoders aren't perfect. We can't actually train this layer in the middle here to only produce single features. We can only train it to produce a sum of features that adds up to 1. And so we might expect some composition happening there. And so we were interested in looking into whether these features are occurring together more often than you would expect by chance. And if that happens, whether this suggests that there is some kind of a compositional logic underlying that feature space. And that might be why the SAE is failing to make these features as independent as we would like. And so that's what I'm going to investigate in my project and what I'm going to talk about in this talk. So basically, the key questions are, how independent are these SAE features really? When are they independent? When are they not independent? How can we interpret the non-independent ones the best? And what is the effect of the size of the SAE on this? Is this a problem that you only get, for example, in small SAEs where you can't make any features and it goes away as we make the SAE larger? So how do I study this? Luckily, this is relatively simple, which is we have this wonderful set of pre-trained SAEs from Neuronpedia. We can download the training data from these SAEs and look through that to count the number of times the SAE features co-occurred in that training set. We then normalize these by Jacquard similarity. I'll explain this more in a second. Convert this into a graph representation, remove those edges that we expect to occur by chance, and then explore the subspaces mapped out by these features. So first, the SAEs from Neuronpedia. I'm going to particularly focus on GPT-2 small and especially on the 8th layer of this model, where there are SAEs on Neuronpedia that are trained at different sizes, going all the way from a very small SAE of 768 neurons, doubling up until I consider the one at 49,000 neurons. So first, we count the co-occurrences. This plot here is just to show features do in fact co-occur. So the blue line here is the fraction of features that are turning on any particular token. We can see that this goes down as the SAE gets larger, as you would expect, but the raw number, this red line here, actually starts to plateau. So you consistently have roughly 30 features firing per token. Now, some of these features are going to fire an awful lot. So here, this is how often a feature occurs, or how many times a feature occurs and how often you see that level of occurrence. And so there are some features that turn on a lot, turn on basically all of the time. And so they would dominate the co-occurrence statistics that we're looking at. So a way to try to control this is we normalize by Jacquard similarity. This is basically designed for exactly this problem, and it just compares the overlap of the set of two features to the total occurrence of the set of two features. And this slightly penalizes cases where you have one feature that's just on all the time, and so everything looks as though it's co-occurring with it because it's just omnipresent. So we convert this to a graph, and the first time we look at this graph, we find we still have a lot of nodes that have a very high degree that seem to be connecting to almost everything, but these tend to have a very low strength, a low edge strength. So they're connecting to almost everything, but they're connecting to almost everything quite randomly. So we remove these edges, and in the Q&A, if people want to ask me why I think that we have this phenomenon, I could go into it a little bit more. But essentially, we remove these very low weight edges, and we see that the graph nicely decomposes into a set of communities. Most of the features end up in a graph of size 1, so they're just firing on their own. But we end up with quite a lot that are roughly sort of size 10 to 20, and a few that are a size roughly 100. And these are the ones that we investigate further. But already, when we just look at these on their own, we find that they look like they're forming reasonable interpretable units. So this is a graph, and by graph I mean a network of nodes that are co-occurring, of SAE features that are co-occurring. So each of these dots is an SAE feature with an ID number and a token that it's promoting. The color here is how often it's occurring in the entire data set, and the edge strength here is how often it co-occurs with different features. And so here we have a set of calendar months that are all co-occurring together, which seems logical that these would come in a single unit. So we then take clusters like this, and we search through the training data for examples of prompts that activate these features, and we perform a principal component analysis of these feature activations to try to see if these features are forming some kind of a group that is more explicable together than they would be alone. So now on to the results. First, we find some interpretable subspaces that, in fact, are kind of reproducing the work we saw with Engels. So again, we find a cluster, for example, of the days of the week. And we find that each of these seems to fire quite often with one particular hub feature that on its own looks like it codes for a Tuesday. If we do a principal component analysis of the activations within examples of cases where these features are on, what we find is that we get these rings again of the days of the week. And just like they saw in Engels, these rings go in the order of the days of the week. But we find a new phenomenon, which is that we have these 3 concentric sets of rings. And the inner and outer set of rings are cases where this token has a space. So just the way in which a machine learning model splits up text is into tokens, and some of those tokens have spaces and some of them don't. And the middle token is the same day of the week, but without a space. So that's a little weird. This is just to show that they go in order. And just to demonstrate what's happening on the level of an individual feature, if we look at just this Friday feature, we can see the places where it activates in this PCA, and we can see that it only activates on this one line in the ring. And so this is actually rather reassuring because it shows that our independent interpretation of what this feature does is exactly what's happening when we look at it as part of this group. And if you want to predict whether or not a token contains Friday, you could just look at this feature. And so it remains nice and independently interpretable. And I'll just say here, when I'm showing a particular case, I will sometimes show a diagram where the color is just the activation of the features in that particular case. And so here's just another view of the same thing. We pick one particular token in the outer part of the ring, and we can see that this Friday feature is very strongly on, and there are a few other features that are on a little bit, but this is the one that seems to be dominating. And this is a consistent pattern across all of these different features. So we have sort of Thursday, Friday, Monday, these sorts of things, except here where we have a little bit of an outlier. And this outlier happens to be, or perhaps predictably, is actually the hub token here. And we can see that this only fires on this inner or middle ring. And that's the ring where we have the difference in this space. So for some reason, this particular feature seems to be when a day of the week is active, it is showing that there is a space in the token. And if we zoom in on the activations in a particular case here, we can see that now there's kind of a splitting of the activity between the feature that is telling you which day it is and this hub feature that is telling you that it is with a space. And this is interesting because if we look back at Neuronpedia, this is the sort of spoke feature. This is clearly a Friday feature. It's not very surprising if we were just looking at kind of the static training data representation. And this one is a Tuesday feature. And the fact that it's having this space effect doesn't seem to be very predictable. The fact that it would do something interesting in composition with this feature doesn't seem to be very predictable. So that's an interesting hint there. And we actually see the exact same situation in months of the year. We have, again, these spokes that go in order. Again, this decomposition into different rings based on whether or not there's a space in the token. And again, the hub feature is the one that is disambiguating between spaced and unspaced cases. So this potentially is a very, very beginning of a hint that there might be some kind of a compositional code happening here with these features that is coming through in the sparse autoencoder, despite the fact that they're not really designed to find that kind of an encoding. So as a next case study, I'd like to talk about classification of something a little bit more ambiguous than days of the week. This is looking at the grammar of the word how. Now we're at layer 8 of the model. So here we have a cluster of 5 different nodes, and here we have a PCA. In a moment, you'll see why it's hard to color this in a way that is obvious, but we already see some kind of a split happening here if we look at the tokens that this is firing on. Blue here is the fact it's firing on the word how. Red, it's firing on the word how, but somewhere nearby there's a question mark. And these all cluster over here. And so there are basically 3 different categories that this set of features is disambiguating. The first is the degree of something. So that's up here in our PCA, and that's encoded by a single feature. And if we look at the tokens that it's firing on in context for a set of points up in this corner, there's lots of different sentences, but I'll save you the trouble of reading them all. They're all cases where you have the word how followed by some kind of an adjective. How difficult this is, how dangerous this, how tough this is. And again, as with the days of the week, this is encoded by basically a single feature that remains relatively independently interpretable on its own. Then down in this corner, we have another case where now it seems to be firing on 2 different features. And these all seem to be followed by a noun or a verb. So how doctors, how customers, how the European Union. But this is a little bit more ambiguous, and perhaps that's a hint as to why it's not able to use a single feature to decompose this. And we have this case that seems a little bit more like the former, where it's how much wetland methane emissions. And that seems to be a little bit more of a degree rather than a manner of something. And so these 2 features fire mostly on this one particular corner. And then finally, we have this one over here, which seems to be cases where how is part of a question. So how did you guys meet? How did you not realize? How did you say that? And again, this seems to be controlled by a single feature. But unlike the days of the week, there's this continuum space in the middle that seems like it would be interesting to explore. And so if we kind of probe different cases along one axis, we can look at the activation of these features either as a bar plot here or just in our little graph of the feature activity. And as we go along, we start out with just one feature being active. And as we move into this ambiguous space, now the other feature that we're heading towards starts to activate. In the middle, we end up with kind of a split between both of them. And then as we end up in the more certain part, we end up, again, having just one feature being active. And this is also going to happen. This is just an animation of the same situation. So here we have the same network, and you can see this movement basically to this question feature from this degree feature. If we go down this side of the PCA, so this is now going from sort of the degree of something to the manner of something. Once again, we see everything moving from one feature to a situation where it's ambiguous, lots of features are firing, and then it ends up at another set of features. So the question that we are left with at this point is, what is happening in these intermediate cases where we have more features active? Are these other features expressing certainty or uncertainty in these unclear cases? Are they helping to disambiguate them? This is something that we want to investigate further. But before we talk more about that, I'm just going to talk about one more case study, which is looking at URLs in Twitter, actually. So here is a set of features that fire on URLs, and they fire on single or double character tokens within that URL. So if I just go to the next slide, here is an example of such a URL. And so it might be firing on just the letter M, or maybe the letter M and the letter Y. And so these are the kinds of cases that we're seeing being picked up. And the colors here represent whether it's a single token or a multiple token activation. You can see that almost all of them are either blue, which is a single token, or red, which is a multiple token. But it gets more interesting if we just focus on the single token activation. So that's what I'm going to do for the rest of this example. And just looking at these single tokens, If we then color by where the token is in the URL, so if we go back to my example up here, here is 0 spaces into the URL after the /. Here's 1, 2, 3, 4, 5. We find that this PCA is disambiguating along this URL. And so these features seem to be measuring how far you are into this URL. And this is a heat map here of the activation of the 5 features in this subgraph, how far along you are in the URL, and then the averaged activity of those features in each of these different cases. And we can begin to see that there seems to be a sort of beginning feature, an end feature, and 2 middle features. And if we look at a sort of area chart of the activity of these features, it becomes a little bit clearer. In the middle, everything seems to be slightly more on, possibly because it's slightly harder to disambiguate sort of a situation. I'm just going to show this as a normalized case. And now I think it's really clear that we have this beginning feature that tails off as you move away from the beginning, an end feature that nicely scales up as you move towards the end, and these middle features that are detecting slightly more accurately where you are. And then this blue feature that is a little bit more mysterious. And if we look at the maximum activating examples on Europedia, it's exactly as we would expect. So this beginning feature we can see activates most strongly on beginning tokens and then tails off. This middle feature is activating on the middle tokens. Here you can see quite nicely it's only activating up until just before the end. And then this end feature activates on the final tokens. Then we have this rather mysterious feature in the middle. This one seems to be activating on tokens that are not stuck together. And in fact, if we look a little bit more closely, it seems to mostly be active on lowercase tokens. Just to show this in another way, here are the same URL now showing the activation of each of these features along this URL. And again, you can see this nice sort of transition from the beginning towards the end. And just coming back to that mysterious other feature, if we look at the number of times it turns on, we can see that it's much more likely to be active on a lowercase token than an uppercase token compared to these other features. So that seems to be what it's doing. But why it would be doing this in the case of this measurement behavior is a little bit mysterious. Does it add anything to this? Why is this clustering so strongly together? Interestingly, if we look at a larger SAE, a more complicated graph emerges. So we have many more features these are doing the same thing. If we plot the PCA, we get again the set of the length that we go through the character coming out. And this blue feature here is again just measuring the lower cases. So it seems to be consistently grouping together with this particular phenomenon. And again, we can nicely see the flow of activity through these different nodes in the subgraph, these different features as we go along. So here, every point that I move in this PCA is going one step further along the URL. And you can see that things basically flow from here into these middle tokens that then end up down here at the end. And this lowercase token just seems to turn on when it's a lowercase token. So again, we're left with an interesting question, which is how accurately are these features actually measuring position? This is a case where there are many more positions that we need to measure than there are SAE features. So how much are we losing resolution when we move to this SAE representation? And if we aren't losing very much, again, that seems to be evidence of some kind of a compositional encoding coming through in the sparse autoencoder, despite the penalty against it being supposed to do that. So finally, I would like to talk a little bit about SAE size. Does this problem or does this phenomenon decrease as you go up in SAE size? In a way, this would be rather nice because having to think about these features in groups is more difficult than having to think about them independently. And we see some hints that things are going to be less important when in terms of this co-occurrence as you go up in size. So this is the size of these subgraphs that we decompose the co-occurrences into as you go up in SAE size. And we can see that the size of these subgraphs decreases. The red line here is excluding subgraphs of size 1, so isolated features that don't seem to co-occur very strongly with anything. And this is including size 1. But in either case, we end up with much smaller subgraphs as we get bigger. The fraction of these isolated features also increases. So this is just the fraction out of the total SAE size of these isolated features. So this goes up, whereas this is the number of activations of features that are occurring in isolated features. So not only is the number of isolated features going up, but their kind of overall importance to understanding what the network is doing potentially is also increasing. So this is all very encouraging. Things seem to be becoming more independent as the SAE size increases. However, if we try to trace a particular cluster that we're interested in as SAE size increases, the picture becomes a little bit more mixed. This is still relatively early. But I pick as a kind of case study here the day of the week feature because this was already seen by Engels et al. It makes a lot of sense. It has this very nice property of the features being arranged in this clock formation. It seems like something that we would like a technique to be able to pull out. And here I try to find that particular property for different SAE sizes. The way I do this is I label our features by whether or not they are activating on a day of the week token. And then for a particular size, I count how many features have this property of being something to do with the day of the week. And I group them into subgraphs. And then I'm showing here the size of a subgraph that contains these day of the week features. And the two colors here are representing how many of the features within that subgraph are days of the week. So basically at 768, we only find roughly one feature that corresponds to a day of the week, as I showed earlier. And it doesn't form a subgraph with other features of the day of the week. It can't. Instead, it forms a subgraph with something unrelated. That's its transparency here. And all the way up to 12,000 nodes or neurons in an SAE, we find that there's an increase in day of the week features. So we're getting more granularity. We're detecting more different days of the week. But they tend to be forming subgraphs that are mixed up with all kinds of other things. At 24,000, however, we suddenly get this subgraph that is very much specifically and only days of the week. And if we perform a PCA on this, it nicely splits into the days of the week as we would expect. And this then persists into the next case, even though we get more features splitting off. And so potentially this feature isn't, or this subgraph doesn't seem to be dissolving as the SAE gets bigger. In fact, it sort of resolves and sticks. This is, of course, very, very early and needs to be looked at with other cases as well. So, future directions. There are lots of questions that still remain from looking at this data set. Some of these are things like, well, why do these features form clusters and some of them don't form clusters? So you get a cluster for days of the week. We don't see a cluster for years in dates. How would we best test whether or not an SAE was learning a local code if the true code was compositional, and how well an SAE that seems to be doing something compositional is actually discovering the real underlying way that the large language model is encoding things? And so are these clusters a good way of extracting these compositional codes, or are they only a very slight beginning? Are the features within these clusters expressing this sort of case of certainty or uncertainty? And how well can we predict the effect of steering on these clusters? That's the next step. Something else that we would like to compare this to is some work by Adam Shai, previously of PIBBSS. He showed that you can recover kind of a representation of how the model is expecting the world to look, a sort of belief state geometry. This seems to be maybe potentially beginning to be related to this question of whether or not the model is trying to disambiguate between different states, and whether we're beginning to see kind of the cases where the model thinks things are uncertain. So there might be some fruitful avenues to compare there. As I hinted at, how much do these features split in and out of these clusters? And how should we think of these clusters as continuous objects when the features underlying them are changing? And there's no real way of saying, well, this feature in a large SAE is the same feature as this one in a small SAE. That's not really how an SAE works. But even more broadly, I'd now like to make a pitch back up to the overall kind of argument of PIBBSS, that all of these problems are not just important in AI safety, but they're also important in biosafety. Today we've talked a lot about what make good features and how can we interpret them. These are the exact same problems that we face in biology, and I feel strongly about this partly because my previous job was in biology. I've been a cancer researcher for the last 10 years. And there we have the same questions. What are the good features? What can we interpret to make predictions? How can we manipulate these? How can we be sure that our manipulations are long-term effective? So in the case of cancer, We used to think that genes were a good feature, but unfortunately they don't seem to be. A case study of this is some recent work by the Martin Carrena lab has shown that even normal tissue that doesn't tend to ever become cancerous, like people's eyelids, contains lots and lots of mutations in lots of really important cancer genes like P53. And so if these cancer genes were nice interpretable features that tell you that whether or not you have cancer, then this should tell you that things are really bad. But this seems to happen all the time in normal tissue. So they're not very good features. So how can we better interpret what clusters of mutations, what context of these mutations are going to lead you to get cancer and lead you to either respond or not respond to treatment? How do we target things such that we can treat the cancer effectively? Again, genes are not good features. We've tried very hard at making targeted monotherapies that affect a particular gene, a particular neuron in a way in this underlying network that cells use. And they don't really work very well. We need to target things in combination, but we don't really understand how to do that very well. And how can we make sure that this kind of steering of the cancer is effective in the long term as the cancer evolves? These are all very similar to problems to one of alignment in a machine learning model. But there's also reason to think that there are going to be similarities in terms of how we might be able to solve these problems. And that's because of the pressures that lead to the kinds of networks that govern cells. So a cell has lots of genes. These genes interact in a network, and that allows the cell to respond to its environment. But the genes within these cellular networks, we can think of these like the neurons in a machine learning model. They don't have the same selection pressure to be a small number. We have lots of organisms that have lots and lots of genes. There doesn't seem to be really an upper limit or a reason to restrict how many genes you have. So we don't expect information to be compressed in the same way it is in a large language model. But there is instead a need for stability. So genes in our cells will mutate all of the time. So you can't have them control a particular behavior very strongly. So an example of this is MYC. This gene is absolutely essential to any kind of wound healing. If there's any proliferation happening in your body, you've probably got this MYC gene active. If you turn it on, what it will do is kill cells. And the reason for this is it codes for both. And the reason it codes for both is because this makes your system more stable, that if you have a single point failure where this gene mutates, then it's not going to instantly cause cancer. So what this means is that core genes that control how cells work are going to be polysemantic. And so we've got a very similar problem to what we've been studying in terms of machine learning models of how do you then decompose this polysemanticity back into something understandable. So we have some similar challenges, but of course, the problem in biology in some ways is harder. We don't know what the connections are very well. We don't know what the weights are, what the logic is. We can begin to try and build models of this, but it's very early days. But I think there is an important reason that people who are interested in AI safety should also have a weather eye on this. One is that there's just an opportunity to transfer machine learning techniques into biology, and that's good for biology. Curing cancer would be quite nice. But also in this biological context, we're going to have a lot of different architectures, a lot of different ways in which these networks process information. And that's going to challenge our mechanistic interpretability frameworks. And that's going to be important for any kind of AI safety of the future, because there's no guarantee that any dangerous AI system that we need to be able to deal with is going to be a transformer-based one. It might be based on a totally different architecture. Here is a case where we have lots and lots of different architectures from nature to practice on. And also because synthetic biology is probably going to be one of the domains that better artificial intelligence is going to lead to rapid advancements in. And so being able to do the same kinds of predictions of, is this system going to be safe? What's it going to do in the long term? How can we best control it will be important there. So to summarize, SAE feature co-occurrence can be used to map interpretable subspaces. The features within these subspaces remain largely independently interpretable, which is great news. It means that SAEs are still a nice, easy to use tool that we can hopefully use to make rapid progress, but they're not completely independently interpretable. And there are lots of cases where it's easier to understand what these features are doing by looking at what features are firing together. This may become less of a problem as the SAE size increases, but there may be some cases where they get stuck and remain in this kind of co-occurrence relationship. And so we need to trace those through. So with that, I would just like to say thank you to my mentor and also people in Lisa, particularly Andy Artiti, who I had very useful conversations with, everybody in the PIBBSS team, and also my colleagues in my previous lab in Jasmine Fisher's lab in UCL, who've been helping me maintain work on some final papers that we're finishing off there. And thanks to basically all of the PIBBSS fellows who've been great. If you want to contact me, here's my email address, and you can also scan this QR code to see my contact details. But with that, thank you for listening.
Thank you very much, Matthew.
Thanks. I see that we may have a question on in the chat. Oh no, it's not. Yeah, so question to attendees. Do the attendees have any questions? They are panelists.
You can put them in the chat or in the Q&A.
Yeah.
Or you can raise your hand if you would like to ask your question in person.
If not, then I will ask a question. What will be like? What is your best guess about what kind of technique from machine learning interpretability might transfer to biology, or maybe even more specifically, the kind of research that you've been doing in some interesting way?
Yeah, I actually think that kind of trying to do something similar to the sparse autoencoder work, splicing these into existing models. So we're just at the very edge of being able to build the kinds of large interconnected network models that truly reflect the large interconnected networks within biology. But then we basically just see, much like in a neural network, lots of things turn on, and then lots of things turn on, and then lots of things turn on, and eventually the cell dies. And you don't really know which things were the most important. What were the determinative factors within there? And I think potentially something as simple as training a sparse autoencoder on the different states as you go through that would make sense. There are difficulties there because, of course, a model or a cell and the models of cells don't decompose into nice individual layers. They don't necessarily have sort of acyclic properties. So there's a lot of difficulties. But that's exactly why I think it's important to have people who are able to kind of look across both domains, because there's a reason why these things have evolved. And it may be that we find convergent evolution again back in the space of computer programs or other things that we're designing at the moment.
Yeah, makes sense. Thanks. Okay, I don't see any more questions, but I guess you are going to be available in the breakout room after all the talks?
Yes.
Okay, cool. So if any questions come to mind in the meantime, you can ask Matthew in about 3 hours, I think.
Yep.
So now I think we are going to have about a 10-minute break.
Yeah. See everyone after 10 minutes.
See you.
See you.